Thank you very much for the invitation. And now I'm talking about life, or you can say universe. You know, universe started with a big bang about 13.8 billion years ago. And the formation of Earth took another 9 billion years, which is like, say, Earth is about 4.4 billion years old. And the first sign of life on Earth came about 3.5 billion years ago in the form of cyanobacteria. And then how basically like the present day life has come up. Darwin actually thought about it, and in one of his notebook, which is notebook number B, which may be his second notebook, page number 96, he drew this diagram. And basically this diagram is the tree of life. His message was all species of life have descended over time from common ancestors. And this branching, like common ancestors, like started with something like here, and this branching pattern of evolution resulted from natural selection. Then by means of natural selection, all different species were formed. And the person basically who gave his entire life on this Darwin's phylogenetic tree of life is Carl Woese. Carl Woese, by that time, by the time he was doing his research, then the DNA technology and DNA fingerprinting and all these things, all these techniques were available. And he took advantage of that, and he basically improved this phylogenetic tree of life. And what I'm doing? And he was the one who first kind of originated the RNA World hypothesis. And this hypothesis was first liked by Walter Gilbert, who got his Nobel Prize in something as in DNA sequencing in 1980. And he supported this RNA World hypothesis for the origin of life based on the concept which was proposed by Carl Woese. Now, what is basically RNA World hypothesis? The RNA World hypothesis basically proposes that RNA was the first central biopolymer in the first life on Earth, and DNA and proteins, these evolved from RNA. And it's because RNA can possess both genetic properties as well as it can act as an enzyme. It can carry genetic information. So it's like a two-in-one situation, one compound doing two things together at the same time. And the people like Sidney Altman, who got his Nobel Prize in 1989, he supported this hypothesis. Based on his experiments, what he did was he took a ribonucleoprotein, and he splitted the protein part and the RNA component from that, and he found out that the RNA by itself is catalytic. It can perform as a catalyst. And then came Thomas Cech, same year Nobel Prize, and he isolated a RNA from a aquatic organism called Tetrahymena thermophila, and found out that the RNA, without the help of any other enzyme or anything else, it can splice into smaller fragments, and these smaller fragments are called ribozymes, and these ribozymes can act as proteins. Okay? Now, our job in RPI basically was to synthesize RNA under prebiotic earth conditions. Okay? Now, in order to do that, what is very important is you need a RNA of length sufficient enough so that it can fold into a three-dimensional figure and perform enzymatic activity. And for that, we figured out that RNA of a chain length containing about 40 to 60 mers are required, because a RNA having that kind of chain length, like 40 to 60 nucleotides, will acquire a triple stamp loop structure like that in order to perform a catalytic activity. And our goal was to see the synthesis of RNA containing a minimum of 40 to 60 monomers, and to synthesize RNA under these conditions. And this was our model reaction. We took a nucleotide, sorry. So we took a nucleotide like here, activated the 5' end, and under neutral conditions at room temperature for about 3 days using clay mineral. It's a particular clay mineral. I'll talk about that later on. And using that, we were able to synthesize RNA, something like 30 to 40 mer. And the clay mineral, the catalysis is very important in this reaction. Without a catalyst, you cannot do these reactions. And the catalyst like we used was mineral, like minerals, clay mineral. And clay minerals are kind of abundant on Earth. And they are basically formed by volcanic eruptions. And one of the clay which we use frequently is called montmorillonite, because first of all it was isolated I discovered in a French town called Montmorillon in southern France. And it's a basically compound with very high surface area, which is like 800 meter per square in 1 gram. And it's a 2-1 layer silicate. It has got different gaps in between. That's where these biomolecules, this is the way actually RNA works. It is a 2-1, sorry. 2-1, we call it bilayer, and that's where that's a gap. It's called interlayer, clay interlayer, and that's where these nucleotides, they are adsorbed, react with each other, as shown in this cartoon over here, and they form RNA. And the way we identify these RNA molecules is by using a technique called high pressure liquid chromatography. And if you can see, like if you start with one nucleotide over here and then add on different nucleotides, you will see that initially you have 2 negative charges in the starting material. Addition of each nucleotide adds up another negative charge to it, and by the time it's like 11 mer, it will have 12 negative charges. And taking advantage of this charge, we can separate these molecules by high pressure liquid chromatography. And but this again is a high pressure liquid chromatography is not a very sensitive technique. So we use another technique called moldy mass spectral analysis, which is right across in this building. And the same compound we can find out, we can detect about 33 MERS. And when we do autoradiography, which is another technique to detect RNA, then the same sample can show the formation of nucleotides up to 55 MERS. And that is basically our goal. And interestingly, this reaction only works for RNA. The difference, like if we use a DNA analog over here instead of RNA, the synthesis doesn't work. So basically it proves our kind of RNA hypothesis that initially it was RNA which was formed and the protein and DNA came afterwards. And it's very interesting to know that these reactions, they operate at a neutral pH. If the pH is highly acidic, it won't work. If it's highly basic, it doesn't work. So neutral pH, which is probably the pH of primitive ocean and all these things, prefer these reactions. And the clay minerals procured from different parts of the world have shown different type of activity. And also, like clay minerals, which we use as catalyst, procured from different depth. Depth basically determines their age. And the clay minerals from different depth or different age also show different catalytic activity. If the clay is too old, then it won't be catalytic. If it's too young, it won't be catalytic. It has to have certain age. And sodium chloride, which is salt, like salinity, is very important for these reactions. Like as shown in this figure over here, at 0 concentration of sodium chloride, reaction doesn't work. and the optimal concentration of salt is about 1.2 molar, and that is the kind of salt concentration primitive ocean also had. So the summary so far is prebiotic RNA synthesis may have been a very simple process requiring only saline clay minerals for the reaction. Okay? The next thing which I'll talk about is chirality. Chirality is basically a signature of life. Amino acids, only L amino acids are used in functional protein, whereas D nucleotides are used for the formation of DNA and RNA. Now, why basically and in case of RNA, you can see these RNA component in ribose component basically in RNA exists in two isomeric forms. One is called D, as another is called L. These are basically mirror image of each other. And only D ribose is present in naturally occurring RNA. And the question is how chiral selection was introduced into the prebiologic system. And in order to work on that, what we did was we synthesized some of the starting material using our chemical methods. So we made D nucleotide of adenine and L nucleotide of adenine plus D nucleotide of uracil and D nucleotide and L nucleotide of uracil 2, mixed them together with clay mineral and observed and analyzed these RNA nucleotides. And the way we did it was like that. We first did some high pressure liquid chromatography of the whole reaction mixture, then just picked up the dimer, for example, I'm showing, and this dimer was further separated and further analyzed using a different kind of HPLC column, and that's where the one product was resolved into so many other isomers over here. Sorry. One product basically, this dimer was separated into that many isomers, and they were kind of quantified, and based on that, we figured out that in our reaction, theoretically, like we should have, when it comes to formation of dimer, theoretically we should have 50% heterochiral products and 50% homochiral products, but in reality, we got preference for, sorry again. So, but in reality, we got preference for the formation of homochiral products. And by the time it was dimer, about 64% RNA products basically were homochiral. And as the chain length grew, the percentage of homochirality increased even up to 97% by the time we got the pentamer. So the conclusion so far was that monomorillonite clay minerals are not only excellent catalyst for prebiotic synthesis of RNA, but they also facilitate chiral selection. And now our present work is to basically see how in a situation like, I would say, chaotic situation like here, where we have all 4 possible nucleotides of RNA and their stereoisomers over here like DL, DL and all these things, when they all together react, what sort of RNA we are going to see. And even if like a dimer is formed, for example, like 2 mononucleotides, they are joined together to form dimer, and these are the number of possibilities. And in reality, only 4 of these will be used in native or I would say in functional RNA. Okay, the rest are not used. So how nature picks up the right products for further like polymerization to form long chain RNA is a kind of subject of our future research. And these research, these studies are also very significant from the point of view of exploration of like Mars, for example. Like the Curiosity mission has so far given us two informations. One information is Mars has got water. And the second information is that Mars is also very rich in these monomorolonite bearing clay minerals. Okay? And now NASA is proposing a rover for 2020 mission. Okay? And the whole idea is to bring the sample back onto Earth. So by say 2022, it's likely that we'll have Mars sample to analyze. And our group has already done some preliminary work and to basically understand how Mars soil or clay sample will look like. And the thing is, like on Earth, these clay minerals, they undergo a lot of changes due to acidity on Earth. And acidity on Earth is due to hydrochloric acid. On Mars is very rich in sulfate minerals. So it's likely that there, those clay minerals will be more kind of affected by sulfuric acid formation, okay? So the clay mineral which we use over here, if they are treated with sulfuric acid, in other words, like if we do a little bit of washing of these clay minerals before using them as a catalyst by mild sulfuric acid, then whether they will retain their catalytic activity or not. So we have shown in our experiments that clay minerals, the terrestrial clay minerals, processed under Martian conditions, when used as a catalyst, they perform as good as clay minerals on Earth. Okay? So that's the message we have so far. And with that, another thing is like when from Mars, when these clay minerals are, they return onto Earth, then they will be distributed in different labs. Okay, and we have figured out that in order, if NASA, for example, is interested in checking those clay minerals from Mars for their catalytic activity, then we will require about 1/10 of milligram for our studies. That's the minimum amount we will require. So that's what it is. So that's where I'll stop. Thank you.